Hello, my name is Madison Long. My senior thesis in the title of my presentation is Monarchs and Milkweed, predicting which milkweed species monarchs prefer. Monarch butterflies are currently listed as an endangered species due to the recent population decline in the United States. This decline is due to the habitat loss and destruction of milkweed, a Asclepias species. Milkweed is the primary plant used for breeding grounds for monarchs to lay eggs and as food for larvae to mature. Monarchs are attracted to the sweet nectars and vibrant colors of milkweed, which makes them an ideal species for breeding purposes. The caterpillars of monarchs use the leaves for food and store secondary metabolites for the leaves as a defense mechanism against birds. Milkweeds are the only plants capable of meeting all of these needs for monarchs. Monarchs migrate every year when the temperatures start to decrease in the United States and Canada. This pattern of migration is known as the monarch corridor. When monarchs migrate, they depend heavily on milkweed because it is the only plant genus that monarchs use for breeding and as larval food. If there's not enough milkweed available, the monarchs will not be able to replenish the current population and produce viable offspring. However, not all species of milkweed are equally beneficial to monarchs. By discovering the traits and defenses of milkweed species, this will help understand which species of milkweed is the most beneficial for the repopulation of monarchs during migration. Knowing which milkweed species attract and benefit monarchs will help in guiding conservation by focusing planting efforts on specific milkweed species most likely to be beneficial to monarchs. My hypothesis for this analysis is that physical traits will have a stronger preference among monarchs when using milkweed species. My thesis is to analyze physical traits and chemical defense mechanisms of many milkweed species to determine how they affect monarch preference. This is how I collected my data. First, I conducted a literature search through ProQuest, ScienceDirect, and Google Scholar to find articles and papers referencing the relationship between any milkweed species and monarch butterflies. I searched words like Asclepias, monarchs, preference, and performance. Papers were included in my analysis if it discussed whether one or more milkweed species were used by monarchs for oviposition, as food for caterpillars, or as a source of nectar via pollination. Then I individually ranked the monarch preference for the milkweed species discussed based on a range from 1 to 3. 1 means monarchs did not interact at all with the milkweed species, and 3 indicated that monarchs had a strong preference for the milkweed species. A score of 2 indicated monarchs would use this species, but they would prefer a species ranked 3 if it were available. This ranking was done for 3 stages of the monarch life cycle: oviposition, caterpillar stage, and adult pollinator. In total, preference data for 16 milkweed species were included in this analysis. In order to discover which traits of milkweed species contributed to interactions with and preference by monarchs, I chose both physical and chemical traits that are known to affect other insect species. I found data on plant height, flower size, distribution, leaf size, leaf shape, and flower color for each milkweed species. For the plant chemical properties, I used a paper by Agrawal et al. In 2008. This paper included information on leaf cardenolides, total phenolics, calmeric acid derivatives, caffeic acid derivatives, flavonoids, glucosin glycosides, and number of phenolic compounds for each milkweed species. Statistical analysis. I used variables that were most important for each monarch life stage. We used a model selection analysis. We ran a stepwise model selection using R. In the stepwise model selection, the dependent variables of oviposition, caterpillar, and pollination were analyzed regarding the independent variables, which were the plant traits and defenses. The plant traits and chemical defenses used in the analysis were numerical, such as height, flower size, leaf cardenolides, total phenolics, calmeric acid derivatives, caffeic acid derivatives, flavonoids, glucosin glycosides, and number of phenolic compounds. We ran 3 model selection analysis, 1 for each of the life stages. In both backward and forward stepwise models to determine which model was best at explaining monarch preferences for oviposition, caterpillar, and pollination stages. In all stages, the backwards model selection gave a lower AIC number and therefore a better model fit compared to the forward model. Therefore, the backward step model selection is the only analysis presented here. A couple of our variables, such as flower color and geographic distribution, was categorical variables, and therefore we could not be included in our stepwise model selection. Instead, conducted two-way one ANOVAs where we tested the geographic distribution and the flower color of the milkweed to see if they affected the monarch preference. To help visualize my model results, we ran a principal component analysis, also known as a PCA, for each monarch life stage using only the explanatory variables that were included in the significant models. These PCAs were completed using R. The first two principal components were then used to visualize both trait vectors and monarch preference scores. Here's a table that contains the 16 milkweed species that was used, along with their ranking, their flower color, their height, and their distribution, just for visualization. In the backward step model selection for oviposition, I found that the best fit model included flower size, plant height, leaf cardinalides, flavonoids, caffeic acid derivatives, fumaric acid derivatives, and total phenolics. However, even the best fit model did not significantly explain the oviposition preference, given that the p-value was 0.1361 and the r-squared value was 0.3731. This suggests that the plant trait included here are not strongly related to the oviposition preference, and a trait or defense that we did not specifically analyze in the study may explain oviposition choices. The 7 plant traits from the best fit model were included in the PCA, where the two principal components explaining about 61% of the variation in this explanatory variables. Monarchs had the strongest preference for taller plants with larger flowers and fewer flavonoids and fumaric acid derivatives for oviposition purposes, though the pattern is not strong since the stepwise model was not significant. In the backward step model selection for caterpillar preference, we found a best fit model that included number of phenolic compounds, flower size, leaf length, leaf cardinalides, glycerin glycosides, total phenolics, flavonoids, caffeic acid derivatives, and fumaric acid derivatives. This model did significantly explain caterpillar preference, since the p-value was 0.03 and the r-squared value was 0.71. Indicating that the model explains approximately 71% of the variation in the caterpillar preference. The 7 plant traits from the best fit model were included in the PCA, where the first two principal components explained about 54% of the variation in the explanatory variables. Therefore, monarchs had a weak preference for milkweed species that were shorter in height and smaller in flower size. The chemical defenses were important for determining whether monarchs will tolerate or prefer a certain milkweed species. Monarchs had a stronger preference for taller plants, larger flowers, more phenolics, and caffeic acid derivatives. The milkweed species that contained these components were highly preferred by monarchs during multiple life stages. For the pollination preference, the backward step model selection found that the best fit model included plant height, total phenolics, leaf length, caffeic acid derivatives, glycerin glycosides, leaf cardinalides, and number of phenolic compounds and flower size. This model significantly explained pollinator preference variation since the p-value was 0.008 and the r-squared value was 0.82 of the variant, and this explained 82% of the variation in the pollinator preference. The 7 plant traits from the best fit model were included in the PCA, which the first two principal components explained 50% of the variation in these explanatory variables. Thus, the monarchs had the strongest preference for taller plants with larger flowers, and with higher total phenolics and caffeic acid derivatives for pollination purposes. Therefore, monarchs had a weak preference for milkweed species with higher leaf cardinalides and other chemical defenses, as well as shorter plant height and smaller flowers. Since the caterpillar and pollinator results are very similar, this indicates a strong correlation in preference among monarchs. Physical traits such as plant height and flower size play a huge role in preference among milkweed species. This is largely due to the surface area needed for caterpillars to thrive in the nectar's initial attraction and the height of the plant for pollinators. Monarchs preference physical traits such as plant height and flower size during pollination, which gives an explanation to our data. This evidence is consistent with our data and thoroughly explains how certain physical traits showed a significant preference for monarchs in certain milkweed species. Between caterpillar and pollination life stages, total phenolics, caffeic acid derivatives, and lower leaf cardinalides have the strongest preference for monarchs. The oviposition model explained approximately 30% of the variation, which is not statistically significant. The caterpillar and pollination models explain approximately 70% and 82% respectively of the variation. Therefore, the caterpillar and pollination models were statistically significant and better model fits compared to the oviposition model. In conclusion, more research should be conducted to increase the sample size of milkweed species, and therefore my hypothesis was incorrect based on the data and the results presented. However, the purpose of this paper is very beneficial in narrowing down traits that monarchs preference in certain milkweed species. The only life stage that was unclear in predicting the preference was oviposition. Therefore, a chemical or physical trait is most likely the explanation for preference in oviposition, which has not yet been analyzed. Based on this analysis, taller and larger plants with more total phenolics and caffeic acid derivatives and less leaf cardinalides are preferred by monarchs during caterpillar and pollination stages. Efforts to restore milkweed populations, milkweed species that favor these attributes are more likely to succeed in the attraction of monarchs through multiple life stages. Therefore, this research is just the first step in discovering the preferences of monarchs and how that directly relates to milkweed species. My references. Thank you.